So it's my great pleasure to introduce our second speaker, Nemanja Vukašinović. Nemanja earned his PhD of Science degree from the University of Belgrade and went on to pursue his PhD at Charles University, where he investigated the role of the exocyst complex in secretion and cell wall biogenesis. He most recently worked as a postdoctoral fellow in Dr. Jenny Resnova's lab at Ghent University. And Nemanja is a plant cell and a molecular biologist, especially enthusiastic about using an advanced microscope to unravel the mechanism of plant development. So today he will be sharing his stunning recent work just published in Cell last month, revealing how polarity-guided mitotic division regulate the plant hormone activity to ensure the optimal root growth and cell states. Nemanja, please.
Thank you, Mingyuan, for the introduction. I'm just going to share my screen now. Everything is fine. I hope you can hear me and see my presentation.
Yeah, I can see it.
All right. One moment. Okay, so thank you, first of all, to the organizers for giving me this opportunity to present my work today. So the work I'm going to present has been done mostly in the lab of Jenny Rusinova at PSB, VIB at Ghent here in Belgium, and together with our collaborators from the Banfi lab and Trevor Nolan's lab now at Caltech. So what we do in Jenny's lab, we study steroids, and then we see where this steroid biology will take us. And today I'm going to show you how it took me to the realm of cell types and cell states. So here I want to emphasize how popular asymmetric cell divisions are in the context of cell types, because asymmetric cell divisions are those divisions that actually give rise to daughter cells with distinct fates, which will then differentiate into different cell types. And this is a recent commentary from the editor of Nature Plants, and here he was emphasizing the importance of Yoda kinase for embryo development and stomata patterning, and he had one disturbing statement which says that symmetric cell divisions are boring because they only maintain the existing tissue and give rise to cells of the same identity. So I have a little mission today to convince you that symmetric cell divisions are actually not boring. And maybe 5 years ago I would agree with the editor of Nature Plants, but today, after all the experience with the project, I would highly disagree. So symmetric cell divisions are most famously studied in root tips, where these proliferative cell divisions produce new blocks that are actually needed for indeterminate root growth. So the central question that many plant developmental biologists are trying to answer is what determines the decision of these cells in the transition zone to actually stop dividing, to exit the cell cycle and go to elongation and later differentiation. And there's almost no metabolic or signaling pathway that does not affect this decision. And in my case, I'm studying it through a lens of steroids. So we're trying to understand how the steroids, which are these steroid hormones, affect this decision. And steroids are famous for having these dwarf type mutants, whether you have it as a mutant of the receptor or a biosynthetic mutant, you end up with these small plants. And the same case is in the roots. So the roots will become short and thick, and this is a consequence of a combination of perturbed proliferation and cell elongation. So steroid pathway is one of the most studied and the best characterized signaling pathways in plants. But for today, we don't need to go through all of these molecular components, and I can show you a simplified scheme with some core players that are important for today's story. So in a hypothetical scenario where we wouldn't have brassinosteroids, the kinases from the ATSK group can enter nucleus. They can phosphorylate BES1 and BZR1 transcription factors, and in this way they will kick them out of the nucleus, and you would have decreased the brassinosteroid response. When the hormone arrives, it's perceived by BRY1 and BAC1 coreceptor, which then leads to deactivation of ATSKases, and then BES1 and BZR1 are dephosphorylated. They can enter nucleus and bind to BR responsive genes. So basically, brassinosteroid pathway is a story of phosphorelays and phosphocascades that eventually lead to this event that BZR1 and BES1 bind to DNA in the nucleus. And we actually use this relocalization of these transcription factors as a readout for the signaling levels. And one of the targets of these transcription factors are brassinosteroid biosynthetic genes. And this is important to note here because this negative feedback loop that they cause is really important to actually maintain optimal hormone levels. So the higher the signaling, the less expression of biosynthetic genes you will get. So the final outcome of BZR1 and BES1 binding to promoters of genes is activation of thousands of genes, which will then lead to changes such as changes in the cell wall structure, which eventually leads to cell elongation, and not necessarily cell expansion, because it's been shown recently that brassinosteroids mainly affect shape of the cell. They by increasing anisotropic growth. So you have these very elongated cells. And in my case, when I joined Jenny's lab, we started looking at the brassinosteroid distribution and movement. And in several past years, we published papers showing that actually expression pattern of brassinosteroid biosynthetic genes is extremely important for optimal root growth, and they're expressed in this elongation zone where we just a second. Yeah, where we actually need brassinosteroids the most because they, as I said, affect cell elongation. But we also demonstrated that brassinosteroids can move from cell to cell through plasmodesmata, which is another level of regulation. And we also demonstrated that there are transporters which are necessary for export of this hormone. And then you would say it's enough. There's so many regulatory mechanisms of brassinosteroid biosynthesis and signaling, but that's not what biology thinks. So it's always more complex than you think. So today I'm actually going to talk about brassinosteroid signaling and biosynthesis in meristems. And meristems are special because in this type of tissue, you have to coordinate cell proliferation with cell elongation. And brassinosteroids are involved in these in both of these processes. So the role of brassinosteroids in proliferating tissues is somehow ambiguous because when you put roots on the brassinosteroids, you will get less cell divisions because cells will differentiate and they will leave proliferation zone too soon. But also when you have not enough of brassinosteroids, again, you're going to get less cell divisions. So because of this, we started hypothesizing actually that maybe brassinosteroid signaling levels fluctuate over the course of the cell cycle, and maybe in different cell phases, cells have different needs for brassinosteroids in order to normally progress through the cell cycle. So in order to study this type of events, you have to include the time axis. Yeah, so you need to study cell divisions at scale, and for this purpose, we used a specialized microscope. Actually, it's a conventional confocal microscope that is just inverted. So then the roots can grow over the normal axis, and what is beautiful about this microscope, it has automatic tracking, so you can put even up to 10 roots in your chamber, mark Meristems, and just go home and come back after 24 or even 48 hours, and you will have plenty of movies that look just like this, and then you can analyze these movies and actually see what's happening with your favorite protein or your favorite gene. So in our case, as I mentioned before, we used BZR1 and its localization. It's actually nuclear accumulation as a readout for brassinosteroid signaling levels. And you can see here a single cell being observed for something like 25 hours, and we can follow from one division to the other and examine what's happening. And we were especially interested in this in these events that happened right before cell division and after cell division. And after quantifying many of these cells, we realized that brassinosteroid signaling, if it is to believe the marker, drops before the division, then it's almost going to zero, and then slowly, actually quite rapidly, not slowly, recovers early in the cell cycle. And when I say early, I mean in the G1 phase of the cell cycle, because we can tell that when we co-expressed BZR1 with the G1 markers. So when we saw this, so first thing we noticed is it was this drop in VR signaling, and we thought what might cause this drop in VR signaling. And I mentioned these ATSK kinases, which are actually negative regulators of brassinosteroid signaling. And when we observed their localization in high temporal resolution, we saw this peculiar pattern. And this pattern is characterized by two things. First one is that these kinases, they flood nucleus right before division or mitosis starts, and then also they remain in the nucleus right after the cell division in these newly formed daughter cells. And this coincides with the absence of the transcription factors from the nucleus. And we actually believe that the nuclear envelope breakdown allows now these kinases to phosphorylate transcription factors that are now exposed to them. There is no nuclear envelope barrier, and this actually leads to a sharp drop in brassinosteroid signaling during cell division. And then, because the drop appeared, we believe that this sharp increase in G1 actually represents a recovery of signaling, which would be needed for normal meristem function. And at this point, we had these observations, but we didn't know if this dynamics of brassinosteroid signaling components had transcriptional consequences. And that's where we teamed up with Latefield Benfey's lab, and specifically with Trevor and Che Wei, who are now at Caltech. And they took a long detour of actually developing a cell cycle reference of the Arabidopsis root. And at the same time, there was a similar activity with a different biological question by Laura at Ken Birnbaum's lab. And they collaborated, and in the end came up with a reference. So now we have two references, so you can choose which one you would like to use. So what Che Wei and Trevor did, they actually produced more single cell samples, and they filtered out proliferating domain from the wild type Columbia roots, and ended up with like 50,000 cells. And after that, they were able to use then they used known cell cycle markers, and they were able to infer this cell cycle pseudo time. So you can see in this heat map some of the examples. And you can also use interactive browser to check your favorite genes to see how they're expressed in the root meristem. So now, once we had this reference, we were able to go back to our original question and ask, is brassinosteroid signaling differentially regulated during the cell cycle? And Che Wei went even further, and he broke down the cell cycle phases in even more subphases, which he named modules, and we were able to see that most of the Braxton steroid regulated genes are actually triggered in this G1 module, which actually corresponds to the imaging that I did. And this is opposite from the cytokinin triggered genes, which are known to be active in mitosis phase, which are actually triggering this G2M module. So now we were confident that what we see actually has biological consequences, and they're not just beautiful patterns of fluorescent tag proteins. And now we said, okay, so if Braxton steroid signaling is decreased during cell division, during mitosis, then we should expect to see that these Braxton steroid biosynthetic genes, which are under the negative feedback loop, also go up early in the cell cycle. And when we examined the expression of the DWAR4 enzyme, which is the first enzyme in biosynthetic pathway, and it's tightly regulated by Braxton steroid signaling, we could see this patchy pattern, which was indication that this gene is indeed cell cycle regulated in cell cycle manner. But what was surprising is that we saw the difference in what we call upper and lower cells. So during these anticlinal divisions, you can actually have upper and lower cell, and we could see that DWAR4 is exclusively expressed in the lower cell after cell divisions, and this happens somewhere later in also in the G1 phase, while we don't have the expression in the lower cell. And this is where things got really interesting, because this was not some kind of self-fate determination, because in every round of division, both cells, upper and lower, would repeat this pattern and give upper and lower cell that have this expression pattern. And that's where we actually come to the term of cell states. So upper and lower daughter cells in these symmetrical cell divisions go through different cell states. One that supposedly has high Braxton steroid signaling and no expression of DWAR4, and the mirroring one, which has high expression of Braxton steroid signaling, Braxton steroid biosynthesis genes and low Braxton steroid signaling. But was that the case? Yeah, so we wanted to know more if this truly is the case. So we started examining Braxton steroid signaling components to see and to actually explain how this might occur. So first, of course, suspect would be BRY1 receptor. We wanted to see if it has differential accumulation, but this was not the case. We didn't see any difference between upper and lower cells in terms of BRY1 accumulation. But then we said, okay, let's go back to actually transcription factors, and let's re-quantify and re-image these roots and see if there is a difference between upper and lower cell. And indeed, there is difference. So in the case of BZR1, we see the pattern that is opposite from DWAR4. So DWAR4 fires in the lower cell, but BZR1 fires in the upper cell. And you can quantify this and see that actually nuclear accumulation of BZR1 is much more rapid and higher in the upper cell. And also, timing is important here. You can see here at the 100-minute mark that DWAR4 is still not differentially accumulated, while BZR1, which is supposedly regulating this gene, is already having this differential accumulation. So then the next question was, okay, let's go even more upstream. So what controls BZR1? And as I mentioned earlier, those are these ATSK kinases. And there are 10 of them in Arabidopsis genome, and we were able to find that one particular one, ATSK32, shows the pattern that we expected. So it's accumulated to more extent in the nucleus of the lower cell, and we could also quantify this. And this is not surprising, because they are negative regulators of Braxton steroid signaling, so you would expect them in the lower cell, where you see expression of Braxton steroid biosynthetic genes. So to summarize this part, at this point, we knew that we have these different cell states, and that the upper cell after cell division, the upper daughter cell, has high Braxton steroid signaling, because BZR1 is nuclearly localized, while in the lower cell, BZR1 is kicked out by ATSKs that can enter nucleus and lower the signaling, and therefore we would have more biosynthesis in the lower cell. And this was the point where we started thinking, we actually wanted to explain how this could be possible. And this is when we started thinking about cell polarity. So it is known that the plant cells, many of them, are intrinsically polarized, which means that there are proteins that can recognize upper and lower membrane of the cell. So in a hypothetical scenario where you would have breast steroid regulator, you would expect that the positive regulators would go up and maybe negative regulators would go to the lower membrane. And then when the cell splits, the upper and lower daughter cells will end up actually with different cargo, let's say, because even though they're symmetrical in morphological terms, they would end up with different physiological environment. So at this point, I really started researching literature, and the family of octopus and octopus-like proteins came as a perfect candidate because they fulfill several important conditions to be considered good candidates. So these genes are known to be expressed in root meristems, and they had even the double mutant of the OPS, and OPL2 had a severe root phenotype. And also, these proteins were already implicated in breast steroid signaling, and they were shown to be positive regulators of breast steroid signaling because overexpression lines mimicked really treatment with breast steroids. And even the mechanism of this phenomenon was known because when octopus was co-expressed with ATScases, in this case BIN2, it was able to drag them out of the nucleus and in this way upregulate breast steroid signaling because they act as scaffolding proteins. So then the last condition remained, and that was the localization of octopus and OPLs. And yes, they localize to the apical plasma membrane, which means that they will be unequally inherited by daughter cells. And if you see here in the co-localization study with B01, you can appreciate that after cell division, octopus or OPL2 in this case is accumulated to a higher extent in the upper cell compared to the lower cell before we can see any differences in B01, indicating that this protein is indeed the regulator of breast steroid signaling. So we of course did more experiments to prove this, such as interactomics and some overexpression studies. But the question remains, so what is this protein actually doing? What is the function of OPLs? And it was actually already shown that they promote so-called meristemness of cells in leaves, where they regulate stomatal patterning. And in our case, we wanted to know if what would happen to the length of the cell cycle if we inducibly overexpress this protein. And we could see that this expression can do two things. First, it can lower the time of the cell cycle or time between cell division to the next cell division. And also, this protein causes cells to divide at smaller size, meaning that they really are positive regulators of the cell cycle. And this is where we constructed our final model. And basically, we think that during the interphase, let's say in S phase, you have some basic or basal signaling levels of breast steroids. And then as you approach mitosis, the levels will drop, partially because ATScases will start entering the nucleus. And this is also a time when some of the octopus or OPLs start to get expressed and slowly go to the upper membrane of the mother cell. And then during mitosis, where the cell plate is still not completely formed, all the octopus will go to the upper membrane and then also to the cell plate. And after cell division, you will end up with cells that has much more octopus in the upper cell. And because they're scaffolding proteins, they will drag out ATScases of the nucleus. B01 will be free to go to the nucleus, while in the lower cell, you will have this moment of time where ATSKases will be able to enter the nucleus and regulate negatively steroid signaling and allow steroid biosynthesis to proceed. In the end, these two cells will equalize. And then you have to ask yourself a question, so why is plant doing this? Why would plant bother to do this complicated mechanism in order to regulate root growth? So the only way to actually test this, or maybe the most convenient way, was to do some computational modeling. And we did this together with the Vabnik's lab in Madrid. And we proposed they already had root growth models, computational models. So we asked them to integrate steroid signaling in these models, and then to try to simulate different scenarios. So for example, scenario where steroid signaling never drops during the cell division, which means that biosynthetic gene expression will never occur. Then in scenario 2, there would be a drop in signaling, and then we would simulate different recovery times. And then in scenario 3, we would simulate what we actually see in plants, that the upper cell quickly recovers steroid signaling and the lower cell slowly recovers steroid signaling, which of course has consequences for the biosynthesis. And what we were able to see in these scenarios is that we repeatedly saw robustness of this scenario number 3, the one that we see in the plant. And these hypothetical scenarios did not behave that well. And one important thing that we also noticed is that scenario 3 could have actually 2 versions, when the upper cell has higher signaling and lower cell has lower signaling, and vice versa. And both of these scenarios behaved in a similar fashion, meaning that what is important is actually just to separate these two cell states. So what we think why plant is doing this is basically to allow coexistence of signal recovery, which sharply dropped during cell division, but also allow steroid synthesis to proceed, which is needed for all other cells in the root meristem. So in summary, I would just remind you basically that, as I said, cell divisions, because of their nature and because of the opening of the nuclear envelope, cause the drop in steroid signaling. This is our interpretation. Then the steroid signaling gets unequally recovered in daughter cells. The upper cell exhibits this high signaling, while the lower one has low signaling and is able to express biosynthetic enzymes. And then this is caused by the polarity of the mother cell, and this is the only way to actually separate the states of these two daughter cells. And the coexistence of these distinct daughter cell types actually circumvents the negative feedback loop and ensures the optimal root growth. And my final conclusion would be that symmetric cell divisions are actually not boring and can be quite fun. So with this, I would like to acknowledge all the people that worked on this project. It really was international and collaborative project. First of all, Trevor and Che Wei from Caltech now, and also the whole team from Benfi's lab. And special thanks to our modeling people, and also big thanks to my PI Jenny and other people from Steroid Group that worked together with me on this project, and the FWO for funding, and of course all of you for your attention. So thank you.
Very impressive and comprehensive work. So unfortunately, we only have time for just one short question. So this is a question from Ramiro. So beautiful results in imaging. So considering the cell type specific signaling of BR in roots, the results you show are in cortex or epidermal cells?
Yeah, so what I showed here is mostly epidermis, but we did play with different cell types, and we know that signaling really is differentially regulated in upper and lower cell in all cell types, but the biosynthesis will depend on the expression of the genes. And wherever the genes are expressed, actually the same pattern occurs.
Okay, thank you, Nemanja. And we actually need to move on to like the central question of our seminar like discussion. So like, if you like we were post all these questions in the Q&A to the Padlet. Feel free to join the conversation there. And I will ask like the question to both of our speakers. So like how could we better define the plant cell types and states in the future? So Jazz or Emmanuel, like which one like to start first?
Well, I was looking back at my old, single cell data from when I was a grad student, and I thought it was interesting. One of the cell types I looked at were natural killer cells, and there was like 10 years before I started doing it, there were basically assumed to be two types of natural killer cells. And then by the time I started working on them, there were people proposed that there were 6,000 to 30,000 types of natural killer cells. So like a huge change in the way people were thinking about them at that time. And I think that it's really important to understand, we can measure these differences and they might be very meaningful, or sometimes maybe it's just kind of random fluctuation and it's not particularly meaningful. So I think the big challenge ahead is to functionally understand what those differences mean, if they do mean anything, or if they're just noise, basically.
Thank you, Jess. Nemanja?
Yeah, I don't know. For me, like I have a feeling like cell types are kind of done. So like what's really challenging is to define the states, and of course all the omics platforms and approaches will be crucial. But since I come from like cell biology developmental background, I think microscopy is also extremely important, and maybe the combo of both worlds will be decisive. And there are already examples from this AI-assisted analysis of root growth from Nakajima lab, which I really admire what they did. And really, already there you can see some things in cellular behaviors that you cannot see with naked eye. So I think the future is in automatic processing and analysis, and then we will see much more.
I think we'll end our discussion like by the last question. So maybe Jazz and Nemanja, can you tell us your definition of cell type and cell state now, based on your study? Maybe Jess, you can. Okay.
I don't know that I feel confident in saying that, but I think there are certainly we tend to think of our data, at least, as meristem versus elongation versus differentiation. I think, though, that is a little bit flawed potentially, because chemically there looks like there's overlaps in certain populations that we wouldn't have expected. But that is at least our first approach, is those cell types and cell states we think about in how they're responding to the environment, and we see differences there as well.
Well, yeah, I think naming stuff is difficult, but I think it's also very important. So I'm quite old-fashioned in this. So I think it's important to name cell types. And for me, this is tightly connected to functionality and the function of the cell. And when it comes to states, yeah, it's more complicated. And I think we have this kind of cyclical states, like with cell cycle or the things I just showed. But you would have this like one direction states in a cell that is differentiating and going through different states. So there I would separate like two type of states. But with types, I would be quite conservative. I think it's important to say this is a phloem cell and this is epidermal cell.
Okay. Yeah. Thank you, Jazz and Nemanja, for the great answers. So I think that'll be the end of this webinar. And feel free to join like the Padlet for more discussion. And Nemanja, there are more questions for you there, so feel free to answer them there. And our next webinar will be on May 5th. So please make sure to join that. another great webinar. And thank you, Joanna, for the coordination. And thank you for all the participants.
Thank you so much, everyone.
Thank you.